want to start now? Or
It's up to you. You can wait. Let's wait a minute.
That's it? Everyone?
I think Chris left his backpack, but I don't know when he's coming back.
Okay, so ready?
Yeah, you can start.
So it's my pleasure to introduce our third and final speaker of this session 4 and the last speaker of the day, Arvind Murugan, who studied first mathematics at Caltech and then physics, and then biology.
As a postdoc, right?
In physics, postdoc, worked in biology, applying what he had learned before. Now is an assistant professor at the University of Chicago. And we're going to hear about doing soft matter.
Right. That's right. Thank you. Well, thanks for that. And thanks to the organizers for having me here. It's a very beautiful place and my first time here. And I must also congratulate you on the wisdom of the collection of talks. It wasn't apparent to me at the beginning of each session, but by the end I realized you guys knew what you were doing. All right. So, so we heard from David and Ting about essentially systems where you can design interactions between some agents. And then the system does something interesting for you. You could call it a computation, some kind of input-output relationship. For David, it was DNA strands, or more abstractly, chemical species with some design interactions, implementing literally truth tables. For Ting, he designed interactions between bacterial species, and then they created different spatial structures depending on initial conditions. I'm sort of going to expand on that, expand the context a bit, by asking if we can use material systems like this to implement a certain larger class, or I don't know if it's a larger class, but a different class of so-called computation or response to the environment. I have in mind things like learning and pattern recognition, which you could call machine learning, or things like control and error correction, things like what Charlie Bennett used to think about, things that you traditionally think of as happening on a computer in software. I want to ask if you can do that kind of computation with sort of interacting systems like interacting material systems, right? And it's going to be a sort of very 30,000-foot level talk. I'm not going to get into too many details, but just all the systems I'm going to give as examples are going to involve networks of some kind. You can model them as networks of some kind, and it's going to be useful for me to have this metaphor, this kind of division, and if you guys have this kind of division in mind. So when you design a network, you can design, sort of, in some sense, clean networks where every component has some role. It's the way we design electrical circuits, and the function of the whole circuit can be traced down to the function of each component here can be well defined, and you know how it affects the total function. On the other hand, you have sort of messy networks, more often found in biology, which can still carry out pretty amazing functions, but usually you can't assign a particular role to a particular neuron. It's somehow the computation is more emergent. So I'm sure most of you have seen this kind of division before, and people say pretty profound things about this kind of division. I'm not really going to, I just need it as an organizational principle for my talk, but it'll be useful if you keep these kind of classes of networks in mind. So I think, Ting was talking about these kinds of circuits that he's designing in the lab, but his real goal, or part of his goal, is to understand the messy networks, and similarly, David as well. There are equivalent systems on the other side. All right, and since I'm going to draw random examples from different things I did, I thought I'd get this over with and tell you the union of all the people who have helped me do the union of all things I'm going to tell you about. So most recently, postdoc Matthew at Chicago, who's been working with me on mechanical systems, Suri on non-equilibrium reaction networks, and James Zhao is like a machine learning superstar, or to-be superstar, that I'm sure you'll hear about. He was a grad student at Harvard when I was working with him. Zorana, a postdoc with Michael Brenner, or used to be a postdoc, now she's a faculty member in Paris, and the senior people I used to work with. All right. With that, we can get to the science. So first, I want to focus on this kind of computation. I told you about two different things. I'm going to first talk about what you could broadly consider machine learning, which is very in vogue these days, especially in California or Boston. But for my purposes, because I'm not interested in software implementations, for my purposes, I'm going to focus on sort of classic samples and ask, can you do the same things with materials? So a classic problem is one of, say, handwriting or speech recognition on a computer. And it's fundamentally a difficult problem for a computer because, the way we write the number 2 can be quite different. And for human beings, it's pretty obvious that these are all numbers at the same numeral. But it's hard to define in terms what kind of variations constitute number 2 and what kind of variations are not number 2 and are number 3 or something. Because in some sense, this object is in a high-dimensional space already with so many pixels, and it's hard to define exactly which ones are this pattern as opposed to that. And for speech, it's even worse. I mean, there are no two people who really sound exactly the same. And in fact, there's a lot of variation in the way we say a given word, and yet the computer needs to somehow classify all words into one and different things and separate different words. So the main inspiration that helped solve this problem, or one way of solving this problem, is this idea of using neural networks, which are in some sense collections of these artificial neurons, which are shown as these nodes. Think of them as some kind of two-state objects. And the way these algorithms work, or these programs work, is you start with a bunch of neurons, which are connected from each other. And there's a training phase in which you show this neural network a typical example of the number 2, or the letter G, or the letter A. And this is called the training phase. When you show it the number 2, some process which adds links between some of these neurons, which means the state of that neuron is determined by the state of the one that's connected to in some way, maybe a stochastic matter, and then the interaction is some kind of nonlinear function. Won't be important for us, but the idea is you first train it, say, with the number 2 or the letter G. It forms some connections, say the blue connections, and then you show it the letter A. It forms another set of connections, say the green connections. And then the claim is, you finish training this network, you take the network and show it some real data, somebody's handwriting in pretty terrible shape. And the network will immediately classify that input signal. Well, these are also meant to be input signals. Will classify those input signals with sort of canonical picture that it's been trained with, right? And so, in some sense, that's how they solve the problem. But the question is how could this work? Just you put in these, neuronal connections, and it's able to solve this difficult problem. When you look at the details, what it really amounts to, what the reason these neural networks are able to do these things, is essentially a designability of landscapes. The thing is, with these neural networks, you can design an arbitrary energy landscape for this neural network, for the dynamical system, which has attractors or deep minima at arbitrary positions. So you showed this letter A, and let's say there's some activity pattern for the neural network which represents A. The claim is you can make the energy landscape have a deep minima at that pattern, have a deep minima at that pattern, and have a deep minima at this other arbitrary pattern. And secondly, when you do that, there's a second requirement, which is satisfied by neural networks, which is that when you scan across all initial conditions, when you show it different kinds of images, there's this very simple attractor structure in the space, in that any initial condition rolls down to one of these programmed attractors, and it doesn't look like this. Namely, the landscape is not actually glassy. Because if you're painting a landscape, you get to paint it with values wherever you want and no values everywhere. But remember, here you only have a limited number of parameters to play with, these neural connections. It's entirely conceivable that when you create these 3 deep local minima, another new minima appears here because you have fewer parameters than the giant landscape. And so the requirement of neural networks, in order for it to function well, this should not happen. So here I drew an example of what can go wrong. You create a new minima, which you didn't intend to put in, which looks like a half G and a half H or something. And these things are bad too, because it means if you show the neural network a signal that sort of looks like an A, but it's not really an A, it might get stuck in one of these glassy things. All right, so those are 2 pretty remarkable conditions. It's hard to imagine that any system would satisfy this. But then there's a third condition, which is even more radical. And it's this condition that in order to create this, that creating this landscape is no more difficult than creating any one of them. The reason is you want this whole process to be, in some sense, learnable. So another way to put it is that I want it, if you tell me the parameters needed to make an attractor at position A, an attractor at position G, and at H individually, I should be able to create this combined landscape by simply, for example, taking the linear sum of those parameters, just taking the union of those connections. The reason for this is that this is nice because what this means is the prescription for combining memories, the prescription for programming in more memories is independent of the content of the memory. So let's say tomorrow you wanted to add a new attractor, a new minima with another letter. You can add it on by just adding more connections on your. I don't need to tell you what has already been put into this network. So hence it's sort of, you can imagine that the system could learn. It could learn A today and then G tomorrow and H the day after, and the physics doesn't need to know what came before, right? Great. I will get to this, yes. Yes.
And number 2 says, oh, they're not going to be minima where you don't.
Right.
What if it makes like a maximum that looks like the negative of the minima you want to introduce when you add that extra thing? Won't they cancel and it'll look, because there's some reason why the non-minimal landscape is flat enough that just adding would work.
Right. Well, that's the requirement. Right. So you want to make sure when you add them up like this, the rest of the landscape is flat enough so this works. Otherwise, it'll be glassy. And that gets to his point. So what I'm saying is these are the requirements. I'm not saying these are the requirements you need in order to have the functionality I described. It's not obvious any system would satisfy it. It wasn't obvious to many people. It was only in the 80s that they realized that this actually can work to some extent, but it's a limited extent, and that's what he was getting to in that. Well, since he let the cat out of the bag, it turns out you can't put in as many as you want. So with a given neural network, this works up to a certain point. You can put in 5 arbitrary attractors. You try to do a 6th or 7th, then this thing happens, and it goes bad. But that was just a long-winded way of explaining what this theory of neural networks is. To me, it's really the most general theory of multi-stability that's learnable and arbitrary. And I want to emphasize that because multi-stability is fairly generic in physical systems. I mean, look, I just picked this random object. This is I'm sure it's multi-stable. Well, I'm sure, yeah, like there. It works, yeah. Yeah, now it's plastic deformation. You just pick a random object with often multiple states. It's not designed. But this is sort of arbitrary multi-stability in that I get to tell you what the global minimum is, and then yet you can create a landscape that has those particular minimum. All right, that was a detour into. Right, so that's a neural network. Yes. Yes.
So a kind of statement is that the patterns you want are local minima.
Yes.
But then a stronger statement is that they really have nice big basins of attraction around them.
Yes.
And
right.
Is the notion, do these two notions give the same number for the capacity? Or?
That's right. In the simplest in all the models I know, yes, but I don't know if so the classic model with sort of what do you call it? Any neuron can be connected to any other neuron, like infinite dimensional neural networks. Those two happen at the same point in that this attractor basin explodes. There is a second transition. Yes.
Yeah, you can't actually do this arbitrarily because as you try to add more minima and the curvatures increase, which would be the vibrational frequencies of the physical system, but as you squeeze more and more minima closer together, the surface has to have a higher curvature, and it's the curvatures that determine the basins of attraction. So there are mutually incompatible things going on here. You can't have large basins of attraction and lots of minima. And this phenomenon has been studied a lot for short-range potentials.
Right.
So for short-range potentials, this actually doesn't care whether this one is there or there or there. You have lots more local minima, you have lots more transition states, and you have higher curvatures. And in a physical system where we've actually modeled this, you can have quantitative analysis of the effect on the global landscape, which becomes globally flatter but locally rougher.
That's exactly right, but this talk is going to be, in some sense, a complement of that, because this thing, so this is sort of magic in comparison to what you just described as physical systems, and the sort of the reason it can promise magic is that the interactions are not short-ranged. So the original example, Hopfield's network, with that and neurons, anyone can be connected to anyone else. In some sense, it's in infinite dimensions. And if you force it down to 2 or 3 dimensions, then actually it has no capacity. You can only put in an order 1 number of memories. So that's why, in some sense, people didn't think about this. So what I'm going to tell you today is how you could do the same thing, but for material systems. But you need to. It doesn't work for most systems because you need this notion of long-range interactions, or you run into the constraints you showed. So I'm going to show you the few examples of material systems which, in some sense, have sufficiently long-range frustrated interactions, which allow this kind of magic in some sense. So this too, this too has a finite capacity. There's a finite number which scales with the size of the system, but that's still much better than the order 1 thing for most physical systems that you just described. Right. So what I want to suggest to you today is that I can change these A, G, or H to be different RNA secondary structures in one RNA strand, or there could be different self-assembled configurations of the same set of particles. It can be literal mechanical folding, or to connect with thing, something I haven't thought about until today. You could imagine the same spatial interactions stabilizing multiple microbial communities, and you choose your community composition and spatial structure depending on initial conditions or external conditions. All right. So I'm going to first give you an example from self-assembly. So a set of self-assembling particles which can be programmed with multiple minima in the sense that we were just talking about. So the context for this is, and the model we made was inspired by, say, these assemblies coming out of the Peng Yin lab. These look very similar to the kind of thing Sean was talking about, but these have no scaffold strand. So these are literally, say, 200 distinct DNA strands coming together, all short, very short strands coming together to assemble some arbitrary structure. And the reason they can do this is because you can create arbitrary specificity in DNA interactions. So what they do is, say, they take 400 different sequences, and they make sure that each, let's say we number those sequences 1 through 400. The sequences apparently fold up into some shape that you can abstract as a square with 4 distinct binding sites. They make sure that the sequence on this side of species 3 will stick to the left-hand side of species 1. So it'll accept only this partner from this side, only that partner from that side, and so on. Right? And then if you let this go, you may or may not be surprised. You should it's at least plausible that it would assemble into something like this, because everything can fit in only one exact way, and they've been able to do it. The real challenges are more kinetic, and so they have to have an annealing protocol and so on. Yes.
If I understand correctly, like in the sort of interior of that heap,
Yes.
those are elements that like accept 4 neighbors and all sides.
Yes.
What sort of determines the size of the interior region?
Oh, I think every little brick here is a unique species. It's a unique particle. So you can number them uniquely from 1 through 400. So it's not some repeating structure. So this is, say, one species.
400 things in solution and let it go, and hopefully it's going to do this.
That's right, and it does, right? And the main difficulty is the fact that you have 400 species. So entropically, there's a big entropic problem, but they got around it to some extent, and they can do that. But the point I want to emphasize here is that what they do is make sure there are highly specific interactions. That 3 doesn't accept some other sequence from that side. Try to minimize that as much as possible, but choosing the sequence is right. And I want to do something that's almost silly. I'm just going to ask, can you take these particles and program in the ability to assemble multiple structures at the same time? Let's say you wanted 3 different target structures. In a sense that I want to have the soup sitting around, and then I want to send in some signal, in the sense of handwriting recognition, and then have it assemble one of these three structures. And I want to do it in a learnable fashion, in a simple fashion, in the sense that all I want to do is make each of these binding sites promiscuous. So to make this structure, these people would have programmed 3 to bind just to 1, but maybe it needs to sit next to 5 to assemble this structure, and next to 8 to assemble the other structure. So I'm just going to make it promiscuous. I'm going to let it accept any partner from that side, 3 partners from that side, and 3 partners from the south side, and so on. So it's a more promiscuous soup. But to make it a little more concrete, let's say the 3 structures I have in mind are all square, right? I have 25 species here. All of these target structures are square, but if you look inside, they're completely different permutations of the same 25 particles. So in some sense, they are like maximally different structures. Even though they all look square, it's like there are 25 people, and if I can get them to arrange in any permutation, it means I can do anything with them, right? And so what am I going to do? I'm just going to take these. It's just simply generated 3 random structures like this, and use that to infer what the interaction should be. So each particle will accept 3 partners from each side based on these 3 permutations, and then we put in, as an initial condition, say, a seed made of 5 particles which were close together in this structure but not close together in the other structures, right? And the question is, will it assemble this? Yes?
I'm not quite understanding the model of what can go. So like looking at the green,
Yeah.
here's 8 and 21 combined.
Yeah. Where?
Over on the left, there's no 8 above 21.
Right. So 8 will bind to 21. So it's an OR condition. So I make them promiscuous, right?
So you say, it forms exactly these 3 structures. It forms at least one more. That 5 by 5 square plus an 8 on top, right?
No, that's my claim. It won't do that. Well, that we're going to see in a moment. I'm just telling you how I prepared the system. It's not obvious if this will work. I mean, depending on the audience, some people think, when I show this today, of course it'll work. Then you put in the seed and you'll get this and not the others, and of course it'll work. And some, especially a biochemistry audience, will say this would never work because these particles are promiscuous.
Well, I'm basically asking, what are the rules for what can bind to what? Because 8 can't always bind to 21, so there's some condition that...
No, that's my whole point. I do not put a condition. I just make, the rule is, if 8 can bind to 21, 8 can bind to whoever it's sitting, 8 can bind to 23, depends on who arrives through diffusion. 8 can bind to whoever, 3 up there.
If you're claiming these are the only 3 structures that can form,
That is my point,
8
yes.
can't bind to that 21 on the left. Otherwise, we do it right now, and there'd be a 4th structure.
You are preempting this.
Sorry.
But that's good. It's good to set it up because it's, right. So to you, it's not obvious this would work. So you're more like a biochemist. Some people, because what you're pointing out is essentially a chemistry argument that this is too promiscuous.
No. I'm
Right.
just asking what's the transition rules for this system.
No, I've already told you the transition rules. You're just telling me it won't work. And that's fair. And that's so what I'm going to call the biochemistry intuition, which is that it won't work. There are some other people who say it's obvious this will work. All right. Anyway, let me just show you what actually happens. So the number of such structures, here there are 3 structures. I'm going to call it M, or M memories. And first we tried this with 5 such structures. We created 5 random structures. And put in a seed corresponding to one of those structures and let it go. Remember, the color shows you which permutation it's assembling. All the structures are made of the same particles, just in different permutations. And you see that it assembles that particular permutation, the red permutation, and stops growing, even though there are still monomers available to glue on. I guess one did glue on, and then we stopped the video, but in general, it stops growing. But that's with 5 structures. Now, we did the same exact experiment, but with 25 structures stored in it, and we're still trying to recover structure number 1. Nothing else has changed. The soup just knows about 24 other structures, and we're trying to assemble the same first structure. And now what happens is what a biochemist would have told you would happen, which is you get giant aggregation. It doesn't stop growing to start with. It grows into an infinite structure made of little pieces of all the different target structures you put in. Yes?
So the rule is that there's no 2 by 2 square in common?
I didn't make any, no, I did not. I just generated random structures and put them in.
They're just random?
They're just random, and it worked for 5. It stopped working for 25, but you're onto something.
Although probably it's true up to some point that there would be no... So in other words, you can always add an additional monomer at the edge because there's going to be something that you can put above a given file.
Right.
But you probably won't be able to complete a 2 by 2 square there.
Right.
There will really be a little isolated notch.
That's right, and it's not even right. And essentially, there's some essentially it depends on the physics of this because there is a chemical potential for these particles, which means somebody attached through one bond is not. So we set the chemical potential to a value such that the bond is not good enough. You need something a bit more than one bond and a little less than two bonds, something between one and two bonds to be stable. That's right, and that is part of the crucial point, and I'm going to get to that in a moment. All right, so you saw that it worked for 5, stopped working for 25, so we just swept it out, right? If you just look at the x-axis here, forget the y-axis. The y-axis is temperature. It's the usual transition, two-dimensional melting. If you look at the x-axis, we just did the same experiment. The experiment is we change the number of target structures, try to recover the same first structure with the seed. You see that it keeps working, it's working, it's working. You program in 14 or 15 structures, all breaks loose, and you get this chimeric mess. I'm going to call this the recovery regime, the chimeric regime. This number 14 depends on the size of the structure you're trying to build, the number of distinct species. So, in some sense, David's intuition is correct here, and essentially what Chris just said as well. Here, this is what David was getting at, that some things can always bind and doesn't work, but there is some window in which it works. And yes?
Are you adjusting your chemical potential? Because presumably at the very low temperature, what you just said to him is not to be true.
At very low temperature. No, we just set the chemical potential to 1.8 bonds in some sense. In this, it works at 1.7. It has to be between 1.5 bonds and 2 bonds, the chemical potential.
Even at the low temperature, a single bond is not stable.
I think the things you're asking about all the mess is between 1 over 10, and there's a reason we stopped this. Yeah, there's all the low temperature stuff that you'd expect, but that's all between 1 over 10, and it's because we cut it off in temperature, not in chemical potential. All right, so why does this work and why does it stop working? And this is essentially Chris preempted this. So I made it promiscuous, so you should expect this to never work because of what David said. The reason it does work is a notion of frustration, and I think I can be quick with this because most of you got this. So let's say these were 3 structures you programmed in. As David pointed out, what's at the boundary here is typically in the interior elsewhere, which means 17 can pick up a partner from this side, 6 can pick up a partner from this side. In fact, M partners from this side and M partners here. M is the number of stored memories. So you should expect this to never work because you assemble your structure, stuff is going to stick on the other side. The reason it might work is that the choices 17 and 6 make are not compatible in the vertical direction, in that they don't make a bond. And you can call that frustration because a friend of a friend is not a friend. And that's frustration. But if you store way too many memories, this is no longer the case. Just by accident, 12 and 28 might just occur in a different structure. And that argument is more of an intuitive argument. The real question is not about these small structures. We were making thermodynamic calculations. The question is, when can you make extended chimeric structures like this? I don't really care if 2 little pieces stick onto my structure. I'm going to ask, when does this go wrong in an extensive manner? You go through that rigorous argument. The scaling law is still the same as this. This square root is because each person in some sense makes 4 bonds. 4 by 2 comes in here. If it was a higher dimensional object or a coordination number, the power goes up. But anyway, the point is there's some window where it works. Here's the other aspect of it. I kept showing you the initial condition as a perfect seed. It doesn't have to be. You could use some imperfect seed, which has got pieces of wrong things glued together with these single bonds that David was talking about. Okay, the dynamics will automatically remove them for you, melt away, and it'll assemble into the structure that's closest to the seed. If this had some blue and some green components, it'll choose most likely the green one because the green one, just the energy, just the dynamics of it. As a result, you can draw out an attractor structure in initial conditions like this for seeds, for arbitrary seeds. But the way we like to phrase it is more as a detector of fluctuation patterns. So imagine you had like n species, a large number of species, and David tells me he wants to detect, he wants a signal, an alarm to go off anytime the concentrations of species 1, 4, 6, and 8 fluctuate upwards together, right? And then Yoav tells me that he cares about this pattern of fluctuations, and someone else tells me that they care about that pattern of fluctuations. This model solves this problem because when you have these fluctuations, these sort of correlated fluctuations, they essentially do nucleation. That's nucleation of a seed, and even if you submit a pattern to it, a new pattern to it, which only sort of resembles this pattern, that's sufficient. That vague pattern will then nucleate a seed corresponding to structure 3, and you'll assemble structure 3. So I want this to remind you of this idea of handwriting recognition. You draw poorly, and yet it gets it. In fact, the math of these two models turns out to be exactly the same thing. All right. I'm going to skip this since many people anticipated it, but this was all worked out. Everything I told you had been worked out before in neuroscience, and as Sid mentioned as well, there is a limit. When you program in less than the critical number of memories, you get a nice non-glassy landscape with just those minima. And then, as you were saying, once you go above capacity, these are the minima. This is from another talk, so the context might be missing here. These are the minima you intended to put in, but because you don't have enough variables, you create all these chimeric minima. The argument is, it's a sort of a flexibility argument like what you were getting at, but this is for systems without certain constraints that physics usually has. All right. They have phase diagrams. I'm going to ignore it. I know this is a talk about kinetics, but I want to leave you with the thing I showed you is not just some accident in self-assembly, but my claim is there are lots of material systems where you have this notion of long-range interactions and frustration, and you have the same behavior. And the one I want to show you, because it's easy to explain, is this idea of design deformation. A lot of people, artists, people making toys, people selling stuff to the army, they take a very rigid object and put in a clever set of just in the right places so that this object as a whole has exactly 1 degree of freedom, right? So you don't need to be careful in how you actuate it. You just go and kick it, and it pops into a hand, or it folds smoothly into a hand because they put the defects in just the right way. And that is great for artistic reasons and for, well, essentially it's easy to deploy, so you need minimal control. Now my claim is, just as for neural networks and self-assembly, if you gave me this program, the red program needed to make a hat, and also somebody else gave me the program needed to make a glove, my claim is I can simply combine them into one object, and that this will controllably fold into this or that. The same problems from the previous time should strike you, because this object is much softer than either of those independent objects because it has the union of both those weak spots. So it could have easily gone completely soft. So sure, it can fold into this, it can fold into that, but it can also fold into many other things if it's very soft, in which case you'd have to micromanage it to get it to fold into any one thing. But that's not the case for a certain class of mechanical objects, and as an illustration, here this is Tomohiro Tachi, an artist, scientist in Japan, who figured out one of these. So it turns out even making individual ones is almost an art now, and it's very difficult to do it. We just came up with an algorithm that can create these individual patterns for you, but Tachi had been doing it sort of in an artistic manner. So Tachi's pattern took a very simple pattern we could come up with, combined them into one sheet of paper. We just laser cut all these hinges in there. And you see, I'm only using 2 fingers. I'm not micromanaging yet, but only the blue creases are folding. Well, that was a little bit of temperature, and now only the pink creases are folding. I don't need to micromanage it to make sure. The non-trivial thing is once some pink creases start folding, only the pink creases get to fold. The blue things don't happen, and vice versa, just like with self-assembly. I don't have time to explain the details of how this works, but some intuition can be gained in this way. So imagine you take this pattern and you crimp this crease. You put your fingers there and fold that crease. When you fold a crease, you need to fold it either as a mountain coming out at you or as a valley going out from you. Right? So let's say you fold it as a valley. It turns out the physics of that vertex will immediately tell the other 3 edges what to do. It may say mountain, valley, mountain. And that signal will propagate down here, up there, and come back and meet itself. The reason the blue thing was hard to design, and Tachi designed it, is the fact that it was cleverly designed so when it comes back, it meets itself and agrees with itself so the whole thing can fold. And because it's difficult to design a single one, it's extremely unlikely that when this deformation tries to propagate into the red pattern, typically it'll meet itself with a contradiction, which means that whole thing has to stay flat. if it tries to fold, it's going to tear because they can't agree on what that crease should do. There's some notion of frustration around loops, just the way I talked about frustration there. It sets another capacity. You keep adding more patterns. At some point it fails. And and so there's a finite capacity again in the same sense. I think I'm going to skip this, but you can play the same game with polymers. The example here is with RNA, but you can think of the DNA origami that Sean showed us today. Usually you take a polymer and design in one set of specific interactions. And the claim here is if you take a polymer and make it extra sticky, give it two sets of interactions at the same time. The claim is that the moment, say, the blue links hook up together, let's say these 2 points come into contact, connected by the blue link, the moment they come into contact, just the three-dimensional constraints of a polymer will prevent the red ones from coming into place. Right? In a statistical sense. I mean, none of the statements I'm making are true or false. It's just statistically it's true that when you put in blue contacts, when the blue staples come into place, the orange staples won't happen, and so you have this partitioning. And somebody actually made this. Somebody made this with RNA 10, 15 years ago. But I'm going to be done with that. And I think with that I want to conclude at least this part, one small part left. But my main idea is when can you find this? The most difficult condition is sufficient frustration. It may not be obvious from what I said, but because I didn't show you examples that did not work. So there are plenty of examples where if there isn't enough frustration, we tried several models, things more like what you said would happen, the capacity would be far smaller.
We don't have a large number of species. Because otherwise,
Yes.
if it was a homoatomic system, it's
No.
promiscuous.
Yes, that's right.
Sees
Everything
everything.
is, it's just aggregation, like what Sid was talking about. It's about large numbers of species. There are other people who have thought about similar things. So Sid's colleague at BU, Pankaj Mehta, with Jim Collins, thought about similar ideas for gene regulatory networks. But I think I'm going to skip past this. But all right. And I guess I wanted to speculate a little bit that, well, today Ting is thinking about very small numbers of species, but as he said, it's a metaphor for, microbiomes and biofilms, which have a large number of species, and there is frustration because, you can have positive or negative interactions between species, and it's high dimensional because the interaction space that's relevant is the species space, so there's no constraint on, how many species a given can interact with. So I just want to speculate that maybe you can design interactions which create multi-stability in this sense, and that depending on the initial conditions, you create different kinds of spatially patterned communities, and maybe each one is adapted to some environment. But that's anyway. So, any questions about that? This was
Yes. I think there's a strong connection between the programming in here and the designability in HP lattice models. So you always work with enumeration of all the possible ways of putting a polymer of a certain length on a cubic lattice.
That's right.
With HP interactions.
That's right.
And then empirically it was found that the more designable it was in terms of the number of
sequences, right?
that gave same energy,
Right.
the lower it was in the lens. So
That's right.
There's clearly a direct connection there with these interactions that you're describing with programmability.
Yeah, so that's what I was trying to get at with the polymer slide, and I think Robin Ball has thought about this. Robin Ball,
Yes.
the person in the UK. Yeah.
Yes. I wonder if you
This was
that's a complete data set. It's
Right.
been fully enumerated.
of HP
I
sequences.
think you could probably analyze it in your terms.
Right, see what kind of attractors. All right. All right, I will think about this. This is more lighthearted, but that was about spatial patterns. But, hedge funds use neural networks to make decisions about whether to buy or not buy. They look at a time series, and the neural network figures out what pattern to look for and says buy based on a pattern. Often it amounts to taking the data series, fitting some internal model. You have some internal model of what the time series is. You fit it, infer parameters, and then based on that decide to buy or sell. And this was interesting to me again because of some work coming out of BU, Pankaj Mehta's group. Do I have time do I have another 5 minutes? And all right. There's this problem where cells need to figure out what the concentration of some external molecule is. And the way you usually do it, here's the external molecule, that red sphere. It binds to a receptor and unbinds stochastically. And so if you're inside the cell, you get some time series like this. The signal is high when it's bound and low when it's unbound. And you want to estimate the concentration of this particle, right? You and I know that the concentration is probably given proportional to the number of times it binds in a given interval of time. That's how much you expect it. But for the longest time, because of biophysical constraints, Berg and Purcell said this is the fundamental limit on how well you can sense the external world. How well can you sense an external concentration? Because they assumed that what a cell could do is measure the average amount of time the receptor is bound to this ligand. So it essentially finds the mean across some interval, total amount of time, tau s. Is that sort of clear what I'm saying? Yes?
So are you going to explain what any of those?
Oh, sorry. Yes. Right. So they assumed.
I don't want to jump ahead.
No. Sorry. No, that was a good question. So this is the error in the concentration. So this is a stochastic process, and the question is, if you try to use the average amount of time the receptor is bound as an estimate of concentration, you can ask what will be the error in that, because you run this experiment many times. Sometimes there's variation because it's a diffusion process, and their estimate of the error was 2 over n, where n is the number of binding events in the observation window. Just the usual, variance is 1 over n business. So the cell observes it for some time t, computes the average of the signal across that time t, and thinks of that as the proximate concentration. And the error would be 2 over n, where n was the number of binding events. And for the longest time, it was thought to be the fundamental limit, until 30 years later, when Wingreen and collaborators at Princeton just thought of this as an abstract mathematical problem and asked, what's the best thing you should compute about this time series to infer the concentration? And it was not the quantity I just told you. It's something else for which I don't have intuition. They just did maximum likelihood estimation. And they showed that actually they'll rate us half of that. The problem was it's some complicated. This I can explain to you right away. This I can't even tell you what it is. It's some property of this time signal. And the question was, how could the cell possibly compute that attribute of this time series, like some other processing of this time series? And so since then, this is Plunkett Methods Group. They came up with complex circuits that does this more complex processing of this time series to infer that parameter. And now we figured it out. I mean, we don't know if it's the real thing in the experiments, but at least we have some models of chemical reactions which can perform maximum likelihood estimation on this time series. Where am I going with this? So what I meant to show with these 2 slides, this is one on circadian clocks, is that there are several biological examples where there's a complex set of reactions which does a complex inference task on a time series. And so far people have worked out like a couple of examples. This one, some property of circadian clocks. And so I wanted to raise the question, two complementary questions. One is just explore this in the sense of David and others. What kind of quantities can you compute about time series? What kind of inference can you do about time series using chemical circuits? And then can you implement those with, say, DNA strands or some other synthetic reactions? It also has relevance for biology because many biologists and others are thinking about how bugs survive in changing conditions. There's some, the environment, let's say, changes between 2 states with certain statistics. Turns out the bug, like some kind of bug, say bacteria, E. coli, needs to switch between 2 phenotypic states at a rate that is on that external switching rate. Or maybe it could perform more complex analysis on that time series. And so this kind of analysis will tell you, how complex a reaction network you need to infer this complex a quantity about the time series. But I think I will stop with that. I was way over time. I'm not going to talk about the last part, but I will stop now. Questions? Yeah.
So I guess about the capacity of your squares.
Right.
So that was a sort of statistical capacity. Throw down numbers.
That's right.
So you could presumably engineer a much higher capacity.
No, converse. Random is the best. So in neural networks, this is well known. Random, it's much easier to program in two things. Say in this case, it's much easier. The reason they could combine these two things was these two structures had nothing to do with each other. There was no structural similarity between them. If there is structural similarity, it gets back to his kind of idea. It's more easy to get them confused. So imagine that these two were two proteins which had made of two domains each, right? And the domains are split at the same. So then if you program both of them in, this part of the sequence could fold into the domain for this protein, and this part would fold into the domain for this protein.
I don't think
yes.
that's what I'm asking.
I guess I'm saying that it's highest when you ask questions about random structures. If it's not random, it means there are correlations, because in some sense randomness means they're not correlated. So if you try to put in if you give me 5 particular structures and ask me to put it in, I may not be able to, even though the capacity is 10, because your 5 structures, you can only do worse than random. So initially they thought of this as a storage device for storing messages or whatever, and the messages you can store best are uncorrelated messages. If there were correlations, they actually would send it through a lookup table. They would first sort of uncorrelate the messages and then store it.
But surely you could add a new fashion that's maximally decorrelated with what already exists.
Yes, but then that would just correspond to the result I'm saying once I put in 14, so in the case with capacity of 14, I'm saying there is no other. I guess my statement is that let's say I've put in let's say I've created a system out here with 13 different things. Maybe my claim is that there exists no pattern. Like there's almost zero probability that there exists another pattern which is completely decorrelated with this. I think that that's what I'm trying to say. All this is limit of large n and all that. It's all thermodynamic argument.
Oh, sure. For log n, I can totally see that. But for this one, how did you design these 14?
Random. Random. Yeah.
But if you think of it like correcting codes, right? If you pick
Yes.
random
Yes.
code
It's better.
works,
Yes.
I mean, okay, yeah, so in the large n limit,
Yes.
it achieves the Shannon limit, but for smaller n,
Yes.
you can do better by
Oh, you're right. Sorry. Yes. Yes. Then I would have. Yes. And this gets to his kind of packing problems. Right. So this is large. Yes.
Problem, there are all sorts of interesting ones that really can't be done, I think, with equivalent particles. So think of trying to design a potential that will stabilize a dodecahedron.
Right.
Particles want to be 3-coordinate. If you start sticking them together, you either get a tetrahedron or a cube. And you don't get a dodecahedron.
How many species are you allowing me?
One.
Right. Right. This is the difference.
Now, I
Right.
can easily design
Right.
a dodecahedron out of DNA.
Right.
If I make all 20 particles different.
Right. That's why I started with the metaphor of DNA, because in some sense, yeah, it would have been silly to talk about any of this if I didn't have the example of DNA. Because, well, you could. It's another metaphor for this. We have our transcription factor complexes, where there are many species of many distinct transcription factors, and the same transcription factors are found in multiple complexes, but in different arrangements. So biology could give you an example. It wouldn't be 400, but might be 20, 30.
The cases that you get for homoatomic systems are always going to be competing morphologies that are both stable, like that 38 atom cluster example.
Right. Yeah, no, this is in some sense the other extreme limit.
Yes.
Many distinct species and designable interactions.
Yeah.
Right.
Well, DNA is inherently programmable.
Right.
That's the whole point, isn't it?
Yes.
Why is your capacity line turning over at a higher temperature? I would have thought maybe,
This one?
yeah, I thought it would go the other way.
Can you really tell where? I can't remember if we actually figured this out. Like, can you tell? I can't tell exactly if this is where the melting line is.
Yeah, I mean,
The solid line.
This one.
solid line. At higher temperature, it's only moving to the left. That's how you've drawn it at least. I would have thought that at higher temperature, I would do a better job of
Removing errors. Again, I think you're thinking of your notion of higher temperatures. I would say is this, and your notion of lower temperatures is stuff not shown here. Here the temperature is getting high enough. Here there's another. What is going on here? I'm a little confused, but yeah. I describe another part. So this is, we simulated sort of real sort of chemical stuff. This spontaneous nucleation that I didn't talk about. So you create the superparticles. I put in the seed, and I'm waiting for this to grow out. But the spontaneously nucleate one of the other memories by accident.
Less frequently at high
That should happen less frequently at high temperature. That is true. Here, it's my point. I mean, his question is, why do I see that here and not down here? Well, I don't know. Oh, I guess the nucleation barrier is higher.
Yeah, the nucleation barrier is higher. Your interactions are weaker.
Right.
It's going to be more rare that you will see.
Right. So it makes sense that you said at low. Wait, what you said is what we see, right?
At high temperature, I expect it would be less probable that I would see spontaneous nucleation.
No, why the opposite?
No, because the probability of making a nucleus gets lower as the temperature goes up, so my particles are less stable.
Right. Oh, wait, what am I saying?
How do you know those are
But,
separately nucleated and not seeded from?
So you only seeded, all the experiments were done, I mean, all the simulations were done with that red seed. So what
Is the red seed fixed, or the red seed can also dissociate?
the red seed, and that's what's happening past this temperature.
Like to think that the higher your temperature, the more likely your nucleus will melt. So this is
Right. Well, what?
The other effect might be that the second cluster
Right.
is just as big, which prevents the.
But we're doing this with grand canonical, there are infinite particles available. So above this line, no stable two-dimensional structure can exist. Mike's question is, so what you see here is that before this red thing got to grow out, some other thing spontaneously, new seed spontaneously appeared, and that obviously happens. What I'm confused by is, should you expect that to happen down here or up here? And he's saying the other way. But
Yeah, something can nucleate at a temperature, and then you make the bonds stronger, a lower temperature, then they're even stronger. You get even more nucleation.
So there's a competing effect. You could denucleate. You could dissociate the seed that you.
Why would that be more likely to happen when they're strong?
At higher temperature, that's what.
No, I'm talking about lower temperature. I'm saying at lower temperature, if something could happen up here, then at the lower temperature, it's even more likely to happen. And that's what exactly. The thing
How did that
here,
hurt you?
you have some kind of very small, like finite supply of particles.
no, it's an infinite, it's grand canonical ensemble.
Okay. But then again, maybe they're all, the bottom ends are growing so fast, you're getting used up.
Oh yeah, it could, that could be.
Again, like I said, I really actually don't know.
Yeah, I mean, we stopped the simulation once it got to this structure, so it's possible, but it still doesn't explain.
It could be that it
The rate-
takes so long to complete the one on the top.
No, but
not the time for it.
yeah, but then Mike will say that the time taken to roll downhill is not as sensitive to temperature as climbing, because Arrhenius thing is exponential in temperature, and what you're describing just running downhill, temperature. Okay, it's been a while. Well, it's only 6 months, but anyway, I won't use the excuse of an infant. Last time I showed this, Mike was in a talk, and he asked me another question which stumped us. And then there was an actual mistake, and we went and fixed it before we published the paper. But now it's published, so I don't know. Anyway. Yeah, as I say, the power is coordination number minus 2 divided by coordination number. So I don't know. I'm not good at this kind of math, but 3 over 4, I would say.
And that's a geometrical dimension, those things are moving. And can you play with the dimensionality on, I said, those particles all like each other, but they also have a pH, or they also have something that, this particle likes these in one dimension, but in another dimension, they can, they have a shape or something. Does that help? With this dimensionality?
Yeah, I guess the way I see it is all those parameters feed in. So this notion of clean capacity wouldn't exist, but yeah, the notion of approximate capacity, and it would be affected by those parameters. You can enlarge it by, say, allosteric interactions, but